The quest to extend lifespan has gone on from the earliest of times. Ancient and modern rulers have employed specialists to find and establish ways to extend their lifespan and rule. The latest rendition of this is biohacking, but that's just a new name for something as old as the hills. Today, we'll focus on the peptide Epithalon, which has been called the immortality peptide. Let's see why. Welcome. We begin our look at the science of aging with a hard biological limit. In 1961, biologist Leonard Hayflick discovered that human cells can only divide roughly 50 to 70 times before they stop, a boundary known as the Hayflick limit. Once cells hit this limit, they enter a state called senescence, meaning they remain in the body, taking up space, but no longer functioning properly. To make matters worse, they spew substances and signals that muck up the function of other cells and tissues both locally and systemically. The accumulation of these worn-out senescent cells is a massive part of what we experience as aging. To understand this limit, we need to look at telomeres. Inside each cell, your chromosomes carry your DNA and are capped at both ends by repetitive DNA sequences called telomeres. Think of them like the plastic tips on a shoelace that protect your genetic information from fraying. However, due to the way DNA replication works, the cell cannot fully copy the very ends of the chromosomes, meaning these protective caps get a little shorter every time a cell divides. When telomeres get critically short, chromosomes become unstable, and the cell stops dividing. Short telomeres are a major biomarker for biological aging, linked to a higher risk of cardiovascular disease, metabolic disorders, and earlier death. Fortunately, an enzyme called telomerase can continuously rebuild telomere length. While telomerase is highly active and reproductive in cancer cells, allowing them to divide indefinitely, it is permanently silenced or turned off in most of our normal somatic cells, like our heart, skin, and lungs. This brings us to a compound developed in Russia over 40 years ago that appears to push back against this cellular limit. Epithalon. Sometimes referred to as the immortality peptide, Epithalon is a simple 4-amino acid sequence: alanine, glutamic acid, aspartic acid, and glycine, AEDG. Moving past the hype of its nickname, the scientific research behind this peptide is genuinely fascinating and represents a molecular approach to aging. Epithalon was developed by Professor Vladimir Kavinson at the St. Petersburg Institute of Bioregulation and Gerontology. In 2003, his team published a landmark study where they treated normal human fetal fibroblast cells, which naturally have silenced telomerase, with Epithalon. The results were remarkable. Telomerase activity was induced, telomere length increased, and the cells exceeded the Hayflick limit, dividing approximately 42% more times than untreated controls. For years, a legitimate scientific limitation was that this research came from a single Russian lab. However, in September 2025, a team led by Sarah Aldemey at Brunel University in London published a peer-reviewed paper in the journal Biogerontology. They independently confirmed that Epithalon increases telomere length in multiple human cell lines through telomerase upregulation. This provided the crucial bridge of independent Western validation for the peptide's core mechanism. Activating telomerase is actually only half of the story. Epithalon was originally derived from an extract of the bovine pineal gland, which acts as the body's master clock. The pineal gland produces melatonin, which is far more than just a sleep aid. It is a potent antioxidant, a mitochondrial protector, and an immune modulator. As we age, pineal function declines, melatonin secretion drops, and our circadian rhythm flattens out, leading to compounded cellular damage over decades. Research in aging animal models shows that treating them with Epithalon normalized their circadian melatonin rhythms, restoring the natural peaks and valleys that flatten with age. Because it appears to stimulate melatonin production, longevity researchers view Epithalon as operating on two synergistic mechanisms simultaneously. First, telomerase activation works to slow the cellular clock. Second, pineal gland restoration works to reset the biological clock. The longevity data in animal models is quite striking. In fruit flies, Epithalon treatment led to meaningful increases in mean lifespan. In mice predisposed to rapid aging and a high risk of cancer, it extended lifespan, reduced tumor incidence, and improved aging biomarkers. Furthermore, in studies on senescent rhesus monkeys, Epithalon was shown to normalize shifted hormonal patterns and restore disturbed neuroendocrine regulation. Despite this compelling data, it is vital to remember that animal models are not humans, and fruit flies and mice do not share our complex aging biology. Currently, we have validated mechanisms in human cells, strong animal longevity data, and human biomarker data showing melatonin restoration, but we lack large-scale, long-term human clinical trials on lifespan extension. The human clinical picture is still developing in real time. Ultimately, Epithalon represents a fundamental shift in how science views aging. Rather than treating aging as an inevitable process, it suggests that aging has molecular mechanisms that may be addressable. While Epithalon is not an FDA-approved cure, it is stable, well-tolerated, and has a 40-year track record. As one of the only compounds ever shown to activate telomerase in normal human somatic cells, it remains one of the most scientifically grounded peptides in the longevity space.